So the next submission is an oral presentation by the NSERC Lake Whitefish Research Program as outlined in CMD 15H 2.116 and 2.116A. I understand that Dr. Wilson will make the presentation. Please proceed.
Good morning. The Lake Whitefish Research Program is an interdisciplinary and collaborative research program based at McMaster University and the University of Regina, and we are supported by funds from the Natural Sciences and Engineering Research Council of Canada through a collaborative research and development grant, and our partner organization in this project is Bruce Power. At McMaster University is my lab. I'm Dr. Joanna Wilson. From the University of Regina, we have Dr. Richard Manzin here with us today, and Dr. Summers unfortunately was not able to be with us, but Ms. Rebecca Eberts is here to speak on his behalf, and a number of our trainees are here with us as well that are directly involved in performing the bulk of the research. Our project has three main aims. The first two are involved at looking at the effects of the cooling water discharge on lake and round whitefish embryos. This is the development period of the embryos that would reflect any animals that are potentially spawning in the nearshore area where the cooling water is discharged. One is to look specifically at elevated temperature and the effects of thermal shocks, and the second is to look at combined stressors, looking at chemical, radiological, and temperature stress in combination on developing fish. Most of our work on these two aims have been on lake whitefish to date. This year we started working on round whitefish. Our third aim is looking at population structure and habitat use from those fish that can be caught in the nearshore area during spawning season, and that's been completed on both lake whitefish and round whitefish from the very beginning. In this map, we show the area where we've done the bulk of our collections, particularly for our population and habitat use studies. We have 8 zones that we've focused on, 2 that we consider reference zones, 1 to the north and to the south of the Bruce Power site, and 6 areas that we consider part of a potentially affected zone. We have sampled fish within these sites during spawning season, using a gill netting approach. We've also deployed temperature loggers as well as embryos back into the field for a number of years and the reference sites and at some sites within the affected zone. That's the first data that I'm going to show you here today. This is data from our temperature loggers from the 2013 to 14 field season. The site in blue, site number 1, is our southern reference site, and you can see that there is lower temperature and much less variability in temperature at that site compared to 2 sites that are near the Bruce A and Bruce B cooling water discharge. And so we see elevated temperature on average around maybe 3 degrees higher than our southern reference site, and more variable temperature in the places where the cooling water discharge is released. And based on mathematical models that we've developed for fish development, we would expect that fish that are developing in those areas impacted by the cooling water, that they may hatch earlier. And this matches what we've done in the field or in the lab with lab rearing experiments, where we don't necessarily see increases in mortality or developmental abnormalities with elevated temperature, but we do see changes in the time of the animals to hatch and their size at hatching. Now, we have done some work on chemical stressors, and in this case, we've focused on morpholine and sodium hypochlorite. Sodium hypochlorite, unless we're at very, very high concentrations way outside the range of anything that would be found in the field, they don't appear to impact our lake whitefish embryos. Morpholine, we can calculate an LD50 that's way above what we would ever expect in the field. We do see some changes in both growth and time to hatch at 10 milligrams per liter. We have also done some work on acute radiological stressors, taking embryos at different points of development and exposing them to short-term radiological exposure. This year, we're still in the process of crunching data where we've exposed embryos to chronic radiological stress throughout their whole development. And we found that early embryos are more sensitive, but that in general, like other fish species, the lake whitefish are fairly insensitive to radiological stress, so it takes quite high doses. We see increases in mortality at 5 to 10 grays. And then we've done some initial studies looking at combined stressors. These are primarily with acute stressors, for example, heat shock and radiation together, or morpholine and radiation together. And while we do see some interactive effects, meaning that effects larger than we might expect based on individual stressor experiments alone, that's only when we reach doses that are far above what's environmentally relevant, suggesting that for the receiving waters, that our single stressor experiments are sufficient to potentially predict effects. Richard?
Dr. Richard Manzin for the record. One focus of my research group is to understand the impact of stressors on embryonic and juvenile whitefish at the cellular level. The heat shock response is perhaps one of the most universal responses to stress, and that's to a wide variety of different stressors. Organisms will produce heat shock proteins, which are fundamentally protective in nature in both stressed and non-stressed conditions. Some of our work to date have shown that in embryos or fry exposed to short-term stress of 3 or 6 degrees above ambient temperature, we do not see a heat shock response. And we only see a heat shock response when we expose embryos to higher temperatures of 9 degrees Celsius. And these are short-term exposures. In contrast, when we expose embryos to repeated 3 and 6 degree heat stress every 3 to 6 days, we do begin to see an elevation in heat shock proteins. But importantly, in these same embryos, we see an attenuated or reduced heat shock response when they are exposed to very extreme heat stress of 12 to 18 degrees, suggesting perhaps some protective effects. Collectively, the data indicate that both embryos and fry are able to initiate a heat stress response, which is protective in nature.
Rebecca Ebert. So as Dr. Wilson said at the beginning, the last theme of our program is to assess the genetic structure and habitat use of fish in this area so that we can understand the impact of our findings in these previous themes. So our goal here is to determine if lake and round whitefish in the affected zone are in some way distinct from fish farther away from this discharge area. So we've assessed this in 2 ways. We've used microsatellite markers to compare genetic distinctness between these 2 areas. And then secondly, we've used stable isotopes to look at whether fish in these areas are distinct in their habitat use. We've done this with fish that we've gill netted between 2010 and 2012, so around 300 individuals of each species. So for our genetic comparison, our microsatellite analysis showed that there is no genetic differentiation between fish in the affected zones and the reference areas. So here I'm reporting the FST values. The closer an FST value is to 1, the larger the genetic distinctness is between 2 groups. You can see that our values for both species are close to 0, implying no genetic distinction. So we conclude that lake and round whitefish in the affected zone are not genetically distinct from those in the reference areas. And then lastly, our comparison of habitat use with stable isotopes, we found that there was high overlap of carbon-13 and nitrogen-15 values for fish from reference areas and affected areas, implying that they're using similar habitat and prey sources. We found that isotopes were quite diverse, implying that these are a diverse group of individuals, possibly reflecting multiple areas of the lake and food webs. But we conclude that lake and round whitefish habitat use is similar and reference in affected areas. So to date, we know that in this 8-zone area, the fish here are genetically similar, and they are also similar in their habitat use. We're now looking to expand this to compare this 8-zone area to populations further away from this discharge area throughout the lake.
Thank you.
Thank you.